Hi, this is Ms. Hodges, and this is the second of the 3 scientific method notes videos. Just to review from the first video, we went over the 3 steps of the scientific method. Identify the problem, where you figure out what you're trying to figure out. Gathering information, that's where you may do a little research, you may tap into prior knowledge prior to making your hypothesis. And step 3, you will make the hypothesis. This video is going to focus on steps 4, 5, and 6. The 4th step is when you have the fun. You will test the hypothesis. This is when you do the investigation, when you actually conduct the experiment. You must follow a set procedure. These are the steps taken to conduct an experiment. They will be on your lab, and you follow each step by step by step, just like you would a recipe. You wouldn't do a recipe out of order, and you wouldn't substitute different ingredients in a recipe. So you do the same with your experiment. You must have constants. These are the factors in the experiment that stay the same. You always use the same brand of material. For example, if you did an M&M lab, you wouldn't compare peanut M&Ms to plain M&Ms. Everyone would have to have the same kind of M&Ms. You always use the same size, weight, or amount of material. You also will have a control. That is the standard used for comparison in an experiment. In an M&M lab, you would refer to the Mars Company's website for their standard colors of M&Ms in a package. They set the standard, and you would compare your packages to that standard. You would have variables, the factors that affect the experiment. First is the independent variable. That is the one that the scientist changes in an experiment. Sometimes I call it the I-dependent variable to help you remember that I, as the scientist, must change this part in the experiment. The dependent variable is the factor being measured in the experiment, and it depends on what you change in the I-dependent variable. Step 5, once you're conducting the experiment, you will record and analyze your data. You need to make and record the observations in a data chart, and data is all the factual information that you find in the experiment. There are 2 types of data. One is quantitative data, and that is measured data. That is number data. When you do a lab and you're taking quantitative data, you're going to have equipment. You'll have rulers, you'll have graduated cylinders, you may have a scale. You are measuring, you're going to measure length and distance, or mass, or volume, or temperature. Think of this as number data. Your data chart will have numbers in it. Qualitative data is descriptive words. You're using the equipment that comes on your body. You use your 5 senses through observation, how it feels, how it looks, how it sounds, how it smells. And yes, taste is one of the 5 senses, but we don't use taste in the lab. It's not safe. This type of data is going to be wonderful adjectives describing what you find in your lab. So your data chart will be full of words and pictures and diagrams. After you have completed, you have to analyze your data. You will graph it. You may make calculations. You may do comparisons, such as Venn diagrams. But you figure out a way to analyze the data so that you can come to a conclusion, which is the final step. The last step of the scientific method is the conclusion. This is where you share everything you discovered with the world. You write a descriptive paragraph explaining what happened in your lab. You start restating the problem, telling them what you were trying to figure out. You restate your hypothesis, what you believed and predicted would occur in the investigation. Then you need to state whether your hypothesis was supported or not supported. Then you discuss and explain the results. You might actually include some of the specific data from your data chart in the paragraph. And at the end, you wrap up your paragraph suggesting ways to improve the experiment, little things that you might need to fix that went wrong. Then you also suggest further ways to investigate the same type of topic for this investigation, so that other scientists can take your information and then replicate your lab or make a lab similar to investigate that science topic. So, the 6 steps of the scientific method. First, you identify the problem. Then you gather information. Then you develop the hypothesis. Then you test the hypothesis. Then you record and analyze the data. And you wrap it all up with a conclusion paragraph explaining your lab and everything you found out.